Anxiety. Among mental illnesses, anxiety disorders are the most common in the United States, where almost 1 in 10 individuals are affected by them. They are more common in women and usually develop before adulthood, but can also develop afterwards. Now, our bodies are heavily interconnected. Absolutely everything we do has impacts on multiple different parts of our bodies. With that in mind, perhaps the anxiety you may feel is not all in your head. There's new evidence that it may also be rooted in your gut. Hi folks, my name is Cole, and I have a Master's of Immunology. Today on Investigate, Explore, Discover, we're going to be looking at the anxious microbiome and therapy. So hang around with me throughout this whole video to get all of the relevant background information so we can dive into some exciting experimental results. Plus, there is more information for you in the description below. Now, state anxiety is something that you and I have felt at many points in our lives. Heck, I get it throughout the process of making these videos. It's defined as a feeling of fear, dread, and uneasiness. It might cause you to sweat, feel restless and tense, and have a rapid heartbeat. By itself, it can be a completely normal reaction to stress. However, when someone exhibits multiple all-consuming symptoms, like being on edge, having a difficulty concentrating, or sleep disturbances continually over 6 months, this is where it becomes something more. It becomes trait anxiety, also known as generalized anxiety disorder. Now, over time, we have identified many ways to help reduce stress and alleviate mood disorders. We can utilize medication, psychotherapy, and self-help techniques, and their effectiveness can vary from person to person. One form of treatment that has gained popularity and proven particularly effective is mindfulness-based cognitive therapy. Mindfulness is an awareness cultivated through meditation and other practices that brings our attention to the current moment and allows us to manage our anxiety as it flares up by assessing the situation without interpretation or judgment. In fact, there are many benefits to exercising mindfulness. It helps to decrease mood disorders such as anxiety and depression, alongside increasing immunity and improving the general quality of life that people experience. If practiced long-term, it can also change the cortex thickness and gray matter density in the brain regions associated with learning, memory, attention processes, and mood regulation. Now, accumulating data have indicated that the gut microbiome can influence brain functions, mood symptoms, and behaviors. Particularly irregular gut bacteria compositions have been observed in individuals with mood disorders such as autism, bipolar disorder, anxiety, and depression. Now, looking at our bacteria is of incredible importance. Though the exact number has been difficult to calculate, there are estimates that there are 1.3 to 10 times more bacteria in our bodies than human cells. Because of the large number of bacteria, they interact in unfathomably complex ways. To try and make sense of this puzzle, we look at gut bacterial populations, usually through people's poop, because we can't directly look inside of people. And there are multiple factors to consider. There's something called alpha diversity, which measures the number of different bacteria types within a single sample. And there's also beta diversity, which measures the similarity of bacterial populations between samples. So groups that look more like one another will group together. Among the bacteria in our guts, we have identified general groups of good and bad bacteria. Probiotic good bacteria, like lactobacilli and actinobacteria, help maintain gut health, while too much bad bacteria like Clostridia and Firmicutes are detrimental to us. Bacteria in our guts break down and metabolize many compounds that we ingest, allowing us to extract energy from our food, producing essential vitamins and allowing the use of medicines. Now, these then get transported throughout our bodies. The metabolized products can interact with other bacteria, our epithelial cells, immune cells, and even our neurons. Now, tryptophan is one of these metabolites. It is known to modulate the immune system, our mood, anxiety, stress responses, and even social behaviors. This is because tryptophan metabolizes into serotonin, which you may recognize as a critical neurotransmitter. Another important metabolite that is processed in our guts is ascorbic acid, also known as vitamin C. Now, it has been shown that vitamin C can reduce stress-related diseases and, of course, helps with immune cell functioning. These and other findings indicate an intimate connection between mental health and the gut microbiome. All of that being said, we still don't know exactly how the gut microbiome influences how effective different cognitive therapies are at treating mental illnesses. Now, I want to take a moment and really highlight why uncovering the link between the gut microbiome and mental health treatments is so important. The gut microbiome is one of the most complex and crucial symbiotic relationships we have. So understanding even a little more about what happens there can enlighten us as to how our body functions. Mindfulness-based interventions are widely effective at alleviating anxiety, but the underlying mechanism is still not well understood. Understanding how the microbiome changes due to mindfulness-based therapies could lead to more effective therapy options. Now, if you also think that these are some important reasons to research this topic, go ahead and tap the like button. This brings us to the paper we're focusing on today. This paper is called Gut Microbiota Associated with Effectiveness and Responsiveness to Mindfulness-Based Cognitive Therapy in Improving Trait Anxiety by Wang et al. From Army Medical University in Chongqing, China. In this paper, the authors aim to investigate the effectiveness of mindfulness-based cognitive therapy intervention in relieving trait anxiety and the changes in the gut microbiota after intervention, and how potential bacteria genera may contribute to therapy responsiveness. For their study, the authors recruited from the medical school students at the Army Medical University and used a fairly stringent set of guidelines, which included that the participants were not on any medications and had not done mindfulness-based therapy before. The 50 young adults that passed this criteria all had a healthy BMI and similar nutrient uptake. These students were given a questionnaire to fill out that assessed their anxiety, depression, mindfulness, resilience, and happiness. Those who scored under 50 on the State Trait Anxiety Inventory were selected to be part of the control group. There were 29 people in this group. The remaining 21 individuals scored 50 or above on the State Trait Anxiety Inventory and were in the therapy receiving anxious group. Compared to the control group, these people had significantly higher levels of anxiety and depression and lower levels of mindfulness and resilience. Interestingly, they never particularly mention happiness, so I guess that's not important for the authors. Either way, there was no significant change between the alpha diversity of bacterial samples among the two groups. But when comparing the types of bacteria between these two groups, they noticed that those who had high and low anxiety scores grouped separately. Now, among the many bacterial species that were present at distinct levels for each group, it was noted that those in the control group had generally a higher level of actinobacteria, which are known to help maintain the mucosal barrier and reduce lipopolysaccharide in the intestine, and those in the anxious group had higher levels of firmicutes, which are involved in energy resorption and potentially related to the development of diabetes and obesity. The anxious individuals then underwent mindfulness-based cognitive therapy treatments, MBCT, for 8 weeks. Gut microbiome and anxiety levels were tracked at the end of it, and 4 weeks post-treatment to assess persistence. Now, the sessions themselves were a weekly 2 to 2.5 hour group-based session, which were a mix of formal and informal troubleshooting and group discussion, which centered around mindfulness principles of awareness, separating yourself from your thoughts, and being present in the moment. Furthermore, around 45 minutes per day of daily home meditation practice was assigned and expected between these sessions. After completing the therapy, those in the anxious group experienced significant improvements in mindfulness and resilience, alongside significant reductions in trait anxiety and depression. Now, to determine the effect of MBCT on the gut microbiome, the authors looked at bacterial populations there. Now, once more, there was no significant change between the alpha diversity among the two groups. But when comparing the types of bacteria between these samples, they noticed that those who underwent therapy had gut bacterium that looked more similar to the control group. What was particularly interesting is that not everyone who went through therapy had the same effects. There were some people who responded very well to it, the high responders, and some people who responded but not as strongly, the low responders. Even more intriguing is that the bacterial populations were distinct between these two groups. Now, many bacterial populations changed as a result of therapy, but the major difference between response rates was a change in subdoligranulum bacteria, which distinctly correlated with responsiveness to therapy differently in each group. Moreover, the low responders had much more of it in their guts, suggesting that the higher level of subdoligranulum, the lower the response rate to mindfulness intervention. Subdoligranulum genus bacteria are part of the Firmicutes phylum, and increased levels are correlated with metabolic improvements in obesity. Of particular note, when comparing levels before and after therapy, subdoligranulum levels did not change significantly between the groups, but they did decrease on average. Now, going a step further, the authors compared the relative abundance of all genes in the bacteria to get a better idea of what MBCT might be doing as a whole. There was a significant enrichment of many genes between the high and low responder groups, which included genes involved in Staph aureus infection, taurine and hypotaurine metabolism, and nitrotoluene degradation. Particularly in the high responder group, there was an enrichment in tryptophan metabolism and ascorbate and alderate metabolism, which metabolizes vitamin C. To quickly summarize everything all together, the authors looked at two groups of people, those with high trait anxiety scores and healthy controls. Those with high anxiety underwent mindfulness-based cognitive therapy for 8 weeks. By going through therapy, this decreased anxiety and generally shifted the microbiome to look more like healthy controls. Among those that went to therapy, there were people that responded better than others, and they had distinct gut microbial populations. Those in the low responders had high levels of subdoligranulum bacteria, while the high responders had bacteria with higher gene expression of tryptophan, ascorbate, and alderate metabolism. Not only do I think that identifying links between the microbiome and mental health is exciting to investigate and learn about, it is also significant in a broader context. This information is significant because it strengthens the proof that there is a biological link between mental health and bacterial populations, indicating that the native composition of the gut can influence how effective a mental health therapy is. Additionally, these findings support that combining gut microbiota therapy with MBCT can possibly strengthen the effectiveness of alleviating trait anxiety. All science is basically a stepping stone for new knowledge, and these steps are driven by questions. And I had a few questions myself after reviewing this study. Now, this study utilizes a relatively small homogeneous sample of young adults where 3/4 of them are men. Furthermore, they failed to collect the parallel control group microbiota after MBCT. So can the data presented here translate across ages, ethnicities, and gender? And does it hold any merit at all? Because there is no parallel comparison. With repetition of this and similar studies, we may be able to find out. Now, the authors are still able to draw conclusions from the work they did. And because this data shows that gut bacterial populations change in response to MBCT, is it possible to solely replicate these results with fecal transplants or through ingestion of probiotics alone? Or would doing these measures only help to ensure the success of therapy, meaning that MBCT is still essential to do? The gut microbiome is known to manipulate tryptophan availability. Tryptophan and its metabolites, like serotonin, are involved in the modulation of many aspects of our health and behaviors. My question here is whether solely increasing tryptophan levels, or particularly serotonin levels, can give the same results found. Though this question is purely hypothetical, because I don't think it's a good idea to really be messing with the neurotransmitters directly. As always though, my final question revolves around you. What sort of ideas or questions popped into your head when hearing about this information? I would love to hear about them in the comment section below. Also, let me know if there are any topics that you'd like to hear about in the future. Ultimately, I hope that you learned something, but more importantly, I hope you enjoyed your time doing so. So if you did, give this video a like and subscribe for more in the future. Well, that's everything for today. Thank you for watching, and I'll see you next time.